Okay, this video recording will be showing you the a new topics, which is the engineered living materials, and this is one paper that discuss the engineered living materials as a topic. So you can also check if you want engineered living materials prospect and challenge for using biological system to direct the assembly of smart materials. So we will go through the some video actually, but I hope you can go directly to the link here, YouTube link, and see what is your impression about that video. I will show you maybe later in the last section. So as an overview, we will go to introductions. So on the left side, this is the top section is the organism. So from this small organism, simple organism, and then human here. This is the complex. Okay, and then the transistor until the AI. So transistor developed for the electronic devices and developed for advanced electronic devices and developed for AI. And here, simple and goes up to plant, animals, and then human. So the more you see from left to right, the more increasing in terms of intelligence. And if you see, or if we observe in details for one cells, the cells actually has a lot of functions already. Okay, so they have the self-regulations, okay, to regulate all the intracellular, for example, the acidity. So how to exchange the ion, the natrium, the hydrogen, and then how to organize their own network here, and then how they are moving, how they are their shape or size changing, and how they self-assembly any actin filament, microtubules. Okay, this is just an overview for cells. So if we see closely the cells, so the cells itself is already comprehensive. They have a lot of functions that we can actually try to replicate in a real world. Okay, so in details, what we are actually discussed from the early class, from the beginning of the class. So we have the stimuli, right? Any environmental stimuli like heat, chemical, light, pH, magnet, electric field, and we have the materials, or in this case, in the biological terms, we have the molecules that have, or that can respond to stimuli, and they have basic functions. Okay, and we have at least three types of functions: practical, regulatory, and analytical functions, and in which they create a complex functions that define intelligence. Okay, so this is a pathway that we want to replicate in developing new materials. So this is examples. Okay, how we change the molecular structure using stimuli, using light, using heat, any formation of bonds or cleavage. Okay, attracting like this, or repulse from here, repulse from here is attracted, and then interacted with the help of visible light or UV. How it can form bonding and cleavage, okay, and how it also form covalent bonding, and also it can be cleavage, and how it's arranged according to the external fields, electric or magnetic, and we can arrange the order, we can adjust the order. Okay, there's a lot of possibility in terms of developing new materials. Okay, in terms of also the how we can replicate to the complex functions in biology. So that's an overview, and I hope you can understand how we are going to develop new materials based on all this overview. So that's why the so we will go to the definition of the engineered living materials. So next generation advanced materials must include smart functional properties that surpass the existing capability. So from this, okay, and also the early slides, so we will have a new standpoint that cells can be considered as nanomaterial factories. And yeah, this is their property, which is the conclusion is they can refashion all these molecules into new structure, into new functions, and they will have a new complex materials, and they can maintain over time. That is the idea why we are using the engineered living materials. And by definition, so this is the definition. They are composed of the living cells that form or assemble the material itself or modulate the functional performance of the material in some manner. So they have this genetic modification for the engineered part, mechanical control, chemical functionalization, biotemplating, and also living as in the cell is biofactory, and they can have the self-healed property, dynamic response, self-growing materials, as in the microorganisms, right? And then for the materials part, they can have the structural materials, biohybrid devices, biocatalytic materials, and biosubstrate materials. Okay, so that's the definition, and then the illustrations we can see here. Okay, the tree is from a single seed, right? The single seed is already have genetic programs or all the information needed, all the essential information needed for growing a tree, from the morphogenesis, how they are self-organized, self-assembly, and how they are maintenance and renewal. And then the engineered living materials, we are using the single agent cells. We are programmed cells, and all the information that we want to have here, we already have it in the cells. So when they are morphogenesis, they are self-organized, self-assembly, they are making their biopolymers, and they will create a new structure that we desire. So that's the idea of the engineered living material. So we are replicate all the characteristics of the tree using the synthetic technology to engineer cells, fabricate, assemble, and maintain autonomously produced materials. Okay. So that's it, the part illustration. So we will go next to the ELMs and composite ELMs. So yeah, these three we have examples for we call it the living materials. So we are using engineered cells and biofilms and use them as living materials, which means we have these polysaccharides, protein structure, and biofilm formations. So this is the example for polysaccharides. How the strategy is. This is just a schematic illustration. There's not much in details here. And then the cellulose, how they are using this biosynthetic pathway and how they recreate the biopolymers so that we can also use the biopolymers. And this is just some examples you can see from the notes here, and I'm not going into details about that. That's all the biologists. Okay, so that's the polysaccharides, and the other is the protein. So the difference is we are recreate some layer of proteins. We can make a template, new template, and we can develop a structured protein. Okay, so that's it. And then the biofilm, we can create a biofilm. We can control by observing the regulation for the genetic cell differentiation, metabolic change. If we can control of that, we can achieve a hierarchical complexity of responsiveness and long-term viability of biological systems. So that's the living materials. So we can use all of these three, so these living materials, and combine them with others. So we can create a new composite, and that composite will have or will consist living materials inside. So we can go to the composite of ELMs. There's a lot of here for the examples. I will go one by one. This is just a swelling and actuation, which is commonly used in hydrogel. So we can use the living materials inside the composites, especially the hydrogel, and this is just an overview how they are swelling and actuated, because they will change in the structure of the hydrogel. Okay. And then the self-lifting robots, this is actually really interesting. They are combining the cells, combining the like here they combining the elastomer, and then they are combining the muscle cells, and then they combine with the optical stimulation to program these robots, whether they can move based on the light. So that's the self-lifting robots. And then they have the smart living surface, which is the surface. Okay, so first we observing the classic non-living material here. We are placing some materials inside, functional materials, and they can release that materials, maybe for drug delivery system or any system that can have this kind of system. And then we have the living surface, so we are changing the functions with the microorganism, and the microorganism, they can react based on their nutrients, and then they can release some byproducts, right? And then this is the interesting part. They can also create the self-cleaning living surface, which is the surface that have, for example, this surface having the fungi. So this fungi will have the nutrients from the food spill. So if your food spilling into this surface, the fungi will clean this food spill, and it will be their food basically. So that's the living surface. And then manufacture polymer matrix. So the microbe will manufacture polymer matrix, which means, like for example here, the bacteria, they can have a reduction process, and they have the polymerization here, and they will template all this polymer, all this monomer, and then they will have a template, and they will have a non-template here. And then when they are separated from the polymer and cells, we have the pattern or templated polymers and non-template. It is useful for creating a pattern or templating polymers based on microorganism. And then the other part is the living carbon materials, which is the combination of carbon and living materials. These living materials can provide metabolic pathway, and then the carbon, they can give additional functionalities. For example, this is the graphene, this is the carbon nanotube, which is they have a good functionality in terms of mechanical property, electrical, and other property that can be used as an advantage. This living carbon materials. And then non-carbon, so maybe this is for metallic and ceramic building blocks. They are used commonly because of the for example, the shells, bones. Okay. And then the bio-inspired silica based biohybrids. This is based on how the cell walls, or the cell walls from different diatom species. Okay, so we can use the silica uh, in to engineer genetically new materials, okay? And then combine them with the other materials as well. So this is the self-healing concrete, using bacteria, using the spores here. Yeah, they are reacting based on reaction because of the air. So if they have a crack, they have air inside, and this bacteria will recreate calcium carbonate, and it is also possible for bricks, and not only for concrete. And the last part is the multi-organism living composite. So you can have two or more than one organism in the composite system. Okay, so yeah, and then it's still developing. It's still new. It's relatively new. I hope we can get more information in the future. So, but now we only have this for composite. And then the last part is the large scale productions. So for example, the first one is tree shaping. We can adjusting the architecture based on tree. Okay, so we can have tree shaping. And then the second one is mycelial materials. We are using the fungi to develop structural materials like this, like chair, for packaging, and how they are actually developed from fungi and the wood feedstock. So they are growing the fungi, and eventually they will kill the fungi and make the form fix. And then bacterial cellulose to recreate the fabrics for clothes, jackets, and the electrospinning of ELMs. So we are using the electrospinning. Basically, electrospinning is for fabricating the fibers. So this is the fibers, but we are incorporating the living materials inside the fibers. And electrospinning is recreating the fibers based on the potential or electricity. So we have the polymer solution. We are injecting through the syringe, and when the polymers release, they will have the electricity or the current, and then they will jump and making a thin fiber. Yeah, that's basically the basic of the electrospinning. But here, the electrospinning, the fiber, they are putting the living materials or the microorganism inside. And this is the 3D printing. Basically, they are using the ink as their microorganism or the living materials, and then they print and making it in a 3D structure. And this is the new one. It's quite complex. It's quite difficult to understand from this picture, actually. So this is the synthesis morphogenesis. We are enabling the complex self-assembly for the ALMs. So if we want to control and program at various scale, we can do that if we design or program at the very beginning for when the cells is having the morphogenesis. So when they are developed into various scales, they will have complexity, and they will recreate the materials by itself. They have the self-assembly property. Okay, yeah, this is really difficult to understand from this picture, but that is what this all about, the synthesis morphogenesis. So that's it for the large scale, and then the conclusion is, yeah, it's really important here. So we are using cells as living nanomaterial factories. So the advantage of using ELM, they are self-assembling, self-healing, and dynamic responsive as the microorganism, right? Similar with the microorganism. And the key for the ELM is they are using biological cells to fabricate, assemble, and maintain materials. And that is one of the key development. And by doing this ELMs, we can could enable the paradigm shift in production of materials. So instead of using traditional manufacture, we can use the cells or microbes as our sources. Okay, so this is the conclusion. So please remember all the key points from this course. I think this is the last part of our course, so I hope you can prepare the homework and the final project very well, and I hope also this this last course can inspire or give you some idea to put into your projects. Okay, so see you next time, and I hope you get a good results for your homework and project. Okay.